Here we go with the answers to the questions for Chapter 5 Critical Thinking Practice. At the first, we started out with an essay. Basically, I wanted you to come up with arguments for or against viruses being alive. Personally, I'm going to take them one at a time and give you my thoughts on the subject. First off, an argument for viruses being alive is that they can take over the metabolism of the host cell. Now, on the not alive side, they have no metabolism outside of the cell. So, are they the perfect parasite, or are they just metabolically active chemicals? That's what we're really asking. You may have put under not alive that they can't live outside of a cell. They have to have a cell to reproduce. Well, in answer to that, there are lots of intracellular parasites that cannot reproduce outside of a cell, and they're not viruses. So, personally, I wouldn't say that was an argument for them not being alive, although lots of researchers go with that one. Here's the kicker. With our current definition, you have to be a cell to be alive. Well, that excludes viruses. They're missing some important parts to be a cell. They don't have a cell membrane, and they don't have, most of them, they don't have ribosomes and the other intracellular organelles to have their own metabolism. So that makes them not alive in the not alive argument. Reproduction. Now, you may have put in a not alive that they can't reproduce by themselves. That is a good argument for them not being alive, but I go back to the lots of intracellular parasites, whereas viruses do make more of themselves. That's usually a characteristic of life. One characteristic of life that they do not have is growth. They do not get bigger after synthesis. All cells get bigger after they're born, so to speak. After a cell divides, the two daughter cells will increase in size until they also divide. Viruses don't do that. They put themselves together like little machines. For the purpose of this exercise, it doesn't matter whether you decided they were alive or not alive. What I wanted you to do is talk about the characteristics of the virus and how it differs from characteristics of cells. On to the next essay question. Is there such a good thing as a good virus? So I've got the no column over here, the yes column over here. Let's take a look at some arguments against and for. They're obligate parasites. Their purpose is death and destruction. Get in, take over the metabolism of the cell, and kill the cell. So that would be in the no category. On the other hand, the friend of my enemy is my friend. So if there's a virus that's killing a human pathogen, I'm going to be liking that virus. There has been research into using bacterial phages that kill, say, streptococcus, and using that as a treatment for strep throat. So the friend of my enemy is my friend. They cause death and disease. Quite a few horrible diseases are caused by viruses, so that would be in the no category. Over here on the yes category, if you are a bacteria and you get infected with a lysogenic phage that carries, antibiotic resistance, that's a benefit to you. The bacterium now has superpowers. So, from the perspective of the bacterium, there are some viruses that you want to get. Mediator of evolution. Under this, what I mean is, viruses weed out the weak, lead to improvement in the overall population. Also, there's been some research into whether viruses have caused big changes in evolution. So, for example, when mammals show up in the fossil record, there's evidence that there was a retrovirus that got inserted into our genome. We still have that virus inserted into our genome. So it's almost like we have lysogenic phages. Now on the no side, they're also the mediator of evolution. If you get eliminated, that's a bad thing. Okay, for this exercise, once again, it didn't matter if you said no or if you said yes, but I did want you to talk about these topics, especially the things that you probably wouldn't have thought about under the yes column. Alright, here's the first multiple choice question. So, if viruses that normally form envelopes were prevented from budding, Would they still be infectious? So our options are 2 no's and 2 yeses. Let's take a look at the no's. Because they are not infectious until they are released from the host. Infectious implies transmission to another host and they are trapped in the host cell. Okay, that sounds pretty good. But let's hop down to this yes. If the host cell were to lyse on its own, the virus would be released and then they could infect other cells. So it looks like between these two we've got to decide which one we like better. Let's take a look at these two. No, because the envelope they acquire through budding has receptors for infecting the next host cell. Oh, there's a good point. We got to think about this one when we're thinking about this one lysing on its own. If it lysed on its own, would it have the receptors, the spikes, for infecting the next host cell? Probably not. Yes, because the host cell is still sick with the virus within it. Infectious. You can be sick without being infectious, without passing microbes to each other. So, personally, I'm looking at this thinking this is the best argument. You got to pick up those receptors. You got to pick up those spikes. So I would say that was the correct answer. This one I would say was close. But what if they're able to get the spikes before they bud out? So I'm going to go with that one being the more correct of the two. On to our next question. If you were involved in developing an antiviral drug, which of the following would be an important trait for effectiveness with few side effects for the patient? So for options we have the drug inhibits a virus-specific enzyme, the drug slows down or stops human cell metabolism, the drug prevents ADS ribosome function, and the drug stimulates the immune system to kill all cells infected with the virus. Well, let's take a look at this one. If a drug inhibits a virus-specific enzyme, it's going to attack just the virus and not the patient. That sounds pretty good. That sounds like a good thing. Slowing down human cell metabolism, that's probably not going to be good, especially if it slows down the immune system. That's not going to help the patient recover very fast. The drug prevents ADS ribosome function. Well, why would that be a problem? Well, your cells have ADS ribosome function, and that would take care of your protein function. Okay, once again, you're going to have side effects. The drug stimulates the immune system to kill all the cells infected with the virus. Well, let's take HIV as an example. Your immune cells, your T cells that help you fight infections are infected with HIV and are being killed by HIV. Well, if your other immune cells come through and wipe out all of those infected T cells, you still have AIDS, whether your immune cells are killed by the virus or by other cells in the immune system. Probably not very good. What if you have a virus infecting your nervous system? That would lead to neuralgia or loss of nerve function. Probably not such a good idea. So I would have to go with a drug that inhibits a virus-specific enzyme. Now, when we talk later about interferons, we will be talking about a drug that prevents ADS ribosome function temporarily. You do have some serious side effects, but they're temporary. Most patients are okay with them. But in a perfect world, we would have just that. Alright. Question 3. Which of the following would be a disadvantage of using bacteriophages, or as we affectionately call them, just phages, to treat human diseases? So this is going back to my example previously about using phages to cure strep throat. Okay, let's take a look at our options. The human immune system doesn't recognize the difference between a phage there to help and a non-enveloped virus there to infect. That would be a problem. Your immune system taking out the phages before they can take out the pathogen. Bacteria are constantly evolving defenses against phages, so we'd need to make new phages as treatments. That's a possibility. Bacteria, other organisms have been known to do that. The phages may mutate and cause infection in humans. That's not terribly likely. Bacteria are so different from humans, their diseases are not our diseases. Some phages produce lysozyme, an enzyme that lyses bacterial cells, which could cause damage to surrounding non-infected cells. Well, if you're causing damage to bacterial cells, probably not going to cause damage to us. especially since, if you remembered from your reading, that lysozyme breaks up peptidoglycan. We don't have peptidoglycan. Our cells are safe. And swallowing or being injected with an infectious agent will never fly with the human public. Well, that is a possibility, but we give vaccines all the time. Probiotics are popular. They're alive. Yeah, so I wouldn't pick that one. So now I have to pick between the human immune system, doesn't recognize the difference between the phage, and bacteria constantly evolving. And personally, I would pick this one first, because I happen to know that when they first discovered phages, they would have strep throat patients gargle with phages specific to that streptococcus, and their immune systems wiped them out. It didn't work. But current studies are able to make them work. I would be curious to find out how. So this one's also a problem, so I would say that that was also a correct answer. So they're both correct. Question 4. People who have lived in or visited Great Britain are not allowed to donate blood in the U.S. Based on what you learned from this chapter about prions, which of the following is most likely reason for this? Prion-caused diseases are slow developing, and the blood donor could be aware that they are infectious. Well, that's definitely a possibility. The American Red Cross is being overly cautious, since prions are transmitted only by ingestion of infected neural tissue. Well, that could be the case, but actually it's good for the American Red Cross to be overly cautious, especially after the HIV, hepatitis B debacle of the 80s, when they weren't screening donors. Prions may travel from the intestinal tract to the brain by way of the blood, making donated blood containing prions infectious. That is a possibility. Prions have been shown to be transmitted through casual contact, so blood donation is definitely out. Well, prions are not transmitted through casual contact, so we can eliminate that one. Prions are infectious proteins, so anything that is high in proteins, such as blood, would be suspect. I wouldn't go with that one. Blood is high in proteins, but you got different proteins all over the place. So it looks like it's between this one. I've decided that the Red Cross isn't being overly cautious, and prions travel from the intestinal tract to the brain by way of the blood, which is a possibility. So I would say the correct answer would be this one. We may have prions in the blood, however temporarily, you can't risk it. And this is also a close second. You inject the DNA or RNA from several different types of viruses into human cells grown in the lab. Now, if this were a proper experiment, one virus per group of cells, if you get my drift. From which would you expect to get more viruses? So we have all of the different kinds of genome that are found in viruses. Double-stranded DNA, single-stranded DNA, double-stranded RNA virus, single-stranded positive sense RNA virus, and single-stranded negative sense RNA virus. So, to answer this question, you need to be able to think through these different genetic types and be able to decide if just the genome is enough to produce more viruses. Let's take a look at single-stranded DNA viruses. Do human cells have enzymes that are made to deal with single-stranded DNA virus? The answer is no. We're built to deal with double-stranded DNA virus because that's what we have in our cells. So we can eliminate that one. Does the human cell have enzymes to deal with double-stranded RNA? And the answer is no, because we don't have that in our cells, so we wouldn't have the enzymes to deal with that. Single-stranded positive sense RNA virus, well, they act like mRNAs. They can go to a ribosome and start making all sorts of viral enzymes. So that's a definite possibility. And single-stranded negative sense RNA. Now, from your reading, hopefully you remember that negative sense RNA have to make a positive sense copy so that that copy can go to the ribosomes. Well, we don't have enzymes to do that in human cells. So the correct answer would be the genomes that are closest to what humans have in their own cells are going to be able to produce more virus if just the genome is put in. So the answer would be double-stranded DNA virus, because that's just like what's in your nucleus, and single-stranded positive sense RNA virus, because that's just like the mRNAs that are coming from the nucleus. So that's it. Remember to email me or get on the Ask the Instructor discussion board if you have any questions about why a particular answer is correct or not.